Damos paso a nuestra primera ponencia que llevará el doctor Robert Wright. Desafortunadamente, el doctor Wright no nos puede acompañar presencialmente por un tema de salud, pero le agradezco enormemente su disponibilidad para hacerlo de manera remota. Muchas gracias.
It looks like Mara's texting me, so I'll assume it's okay. So, I'm very sorry to not be there in person. I had an unexpected health issue that prevented me from traveling, but I am very honored to be here today for Dr. Mara Diaz-Rojo, who's been my friend and research partner for about 25 years now. So we started the Progress cohort together. Our first grant was funded in 2006, but the work really preceded that by several years. And much of that actually led to what I'm going to talk about today is because, as Mara said, the Progress cohort is actually designed as an exposome cohort. I'm going to propose an idea rather than present data. And that idea is that a human exposome map, and by that I mean a geospatial temporal map, not a biological map, is actually the best way forward for exposomics and really the grand scientific challenge of the 21st century. Also, because I like to put in a shameless plug for my blog, I write a blog on exposomics, which you can see at this link here, but you can also just Google my name and exposome. Okay, so I wanted to start with a couple of photos of Mara. As I said, we've known each other for 25 years. We've gone on trips together with Salvador and her children, Andres and Inez, and that's my wife, Roz, next to Mara on the left there when she visited New York, actually during COVID, and we had dinner in Times Square. We've had many events in Mexico. So they've always been wonderful and felt welcoming. So what is exposomics? So just to make sure that everyone is clear on what we mean by that, the term itself was first described by Dr. Chris Wild at IARC, International Agency for Research in Cancer, and he defined it as all environmental exposures from conception to death. So why do we need a map? Well, I think if we have an exposome map, we can actually start to understand the exposome, both past and present, and that we can learn lessons actually from what genomics did. So genomics learned about the genome's function and structure from mapping it. And I propose that we can accelerate our understanding of the exposome by mapping it as well. So this is really the why of exposomics. So there's at least two aspects of exposomics that appear to be intractable. The first is what everybody understands, that you're trying to measure all the different types of environment, social, chemical, physical, and biological, all at once. That's an easy concept to grasp. But what people seldom think about or consider is that the other aspect of the exposome is that we're trying to measure all the environment from conception to death. And that's actually even harder than measuring all environment cross-sectionally or all at once. In order to do this, we have to reconstruct the past exposome, as almost all human research starts after a person is conceived. There are no good studies that go all the way from conception to death. And yet this is really critical to human health. In fact, no less than Francis Collins, the former director of NIH, at his exit interview with NPR said, "Your likelihood of having a certain lifespan depends heavily on the zip code where you were born, and that is a reflection of environmental exposure, socioeconomics, social determinants of health, etc." So even Francis Collins, a geneticist, acknowledged that where you're born or geospatial understanding of your environment is very, very critical to health. In fact, it's a better predictor of health than DNA sequence. So why is the past exposome so important? Well, many of us are familiar with the developmental origins of health and disease, that biological and environmental events from our early life predict our current health. This was first described by David Barker, and it's sometimes called the Barker hypothesis. In order to understand our early life, however, it requires being able to go back in time. For example, if we want to understand the developmental origins of Alzheimer's disease, we could either start a priori and enroll children right now and wait follow them for 80 years until they develop Alzheimer's disease, some of them and those that don't, and then look back at their exposures to see which ones predicted it. Well, everyone in this room would not live to see the end of that study. So our only hope of understanding the developmental origins of many adult diseases is to be able to reconstruct the past. And maybe that's possible on a grand scale. So I'm going to start with an analogy. So I was a literature major in college, and I'm very fond of magical realism. And so also this is an event taking place in Mexico. So I think it'll be an audience very familiar with magical realism as a genre. So Jorge Luis Borges is one of my favorite authors, and he wrote a short story about a man named Ireneo Funes who had a perfect memory, and how remembering everything, everywhere, all at once, all the time drove him mad. So here are some excerpts from the story. Funes remembered not only every leaf of every tree and every patch of forest, but every time he had perceived or imagined that leaf. It irritated him that the dog of 3:14 in the afternoon, seen in profile, should be indicated by the same noun as the dog of 3:15, seen frontally. To him, each memory of the dog was unique, not just the dog itself. Funes had no insights on life, only facts that he could recite in minute detail. What Borges was really talking about was a type of philosophy called nominalism. Nominalism is different than the prevailing philosophy of the day, which is realism. So in realism, which was the philosophy of Plato and Aristotle, the world was defined by particulars and universals. Particulars share characteristics of the universals. Cody is a dog, Frodo is a dog, Frodo and Cody are particulars, dog is a universal in this example. In nominalism, there are no universals. There's only particulars. This is very Machiavellian, so there is no good or evil. Each behavior and action is unique. Cody is Cody, Frodo is Frodo, there is no dog. Taken to its extreme, Cody this morning is different from Cody who's playing with Frodo now. That's a nominalist approach to philosophy. I'm going to argue that exposomics is in danger of becoming a nominalist science. It's because we're expanding it constantly and we're just measuring things. We're not really understanding their structure, their correlational structure, not just their biology, but also how they covariate. So the exposome is composed of every exposure to which an individual is subjected from conception to death. That was Christopher Wilde's definition. He included internal processes, metabolism, hormones, the microbiome, inflammation, external exposures, radiation, infections, air pollution, socioeconomic, psychological influences, social capital, education, etc., mental stress. And then this is all superimposed on exposure timing because life stage specific effects occur as well. So exposomics, I would argue, is in danger of becoming a anomalous science by trying to measure everything, everywhere, all at once, and all the time. Like Funes, if we proceed this way, we're going to catalog the exposome without really understanding how it operates. I would like to propose that we redirect exposomics towards realism. Instead of trying to measure everything, perhaps our goal should be to impute the exposome by defining its correlation structure and interrelationships. If we can do that, all we need to do is measure a subset, and then we can model the rest. More importantly, this can help us reconstruct the past exposome. And we can do this by anchoring the physical, chemical, social, and biological exposomes together with geography and time using maps. So how? Well, we can apply lessons that genomics learned to exposomics. Chromosomes are the genome's geography. The genome map showed that genetic variants that were close to each other on chromosomes tended to be inherited together. Regions of linked variants became known as haplotypes. This led to the haplotype map, or the HAP map. This took advantage of how single nucleotide polymorphisms, or SNPs, and other variants are organized geospatially to efficiently conduct genome-wide association studies. So genome-wide association studies are typically done on a subset of SNPs that are used to predict the presence of other SNPs using the correlational structure of the genome. Essentially, genomics was exploiting geographical correlation in the genome landscape in order to accelerate research. And in fact, even with the advent of whole genome sequencing, Many studies actually prefer to use GWAS because it's cheaper and it's more efficient. So early genomic maps went from two-dimensional, which this is, which were relatively primitive, to three-dimensional, and now maps that can actually estimate the conformation of the genome at different times, that are incorporating aspects of epigenomics, for example, and that can help to predict transcription. So these maps started out primitive, but became more and more advanced. And how were they used in GWAS? So the genome map revealed haplotypes. And as I said, these are SNPs that are highly correlated. So if you had a set of 4 SNPs on a chromosome, big A, little b, big C, little d, and then you're also little a, big B, little c, and little d, they tended to be inherited together. So if you knew your genotype for B, whether it's big B or little b, you would know the other SNPs that got inherited with it. You don't have to measure them. If you have little b, then you have big A, big C, and big D because they are close together and they tend to be inherited together. These were called haplotype tagging SNPs. A tagging SNP predicts the presence of other SNPs in a haplotype block. Associations between the tagging SNP and the phenotype represent an association with the chromosomal region, so a geographic space on the chromosome. Today, reference genomes are actually used to impute unmeasured genotypes after a subset of SNPs are measured. So this got expanded. And instead of having a small number of haplotypes that used as the reference, there are now thousands of reference genomes that are used that are actually correlated to the person that you're actually genotyping so that you can estimate the other genotypes more accurately. We could do this in exposomics. So exposome maps are temporal as well as geospatial. All of our air pollution and climate models go back in time. Everything on a map can reflect calendar dates and time. If we map chemical biomarkers and we log the time and place in which that person lived, we could also go back in time. So we could create life course temporal spatial exposome maps that actually change with time. By this analogy, if we map exposure biomarkers, each person is like an EPA air monitor in an air pollution study. And we could even map chemical mixtures using commonly used data visualization techniques so that we could actually break it down by life stage, whether it's a child or an adult or calendar year, whether it's the 1980s or the year 2000 to 220. We could see both the temporal variation in these mixtures, but also the life stage variation in these mixtures. And we could co-locate other aspects of the exposome, such as income and other chemicals, as well as health outcomes using AI and machine learning. So we could look at geolocation of these different environmental exposures and whether they correlate with hotspots of disease. First, we need to define a nomenclature, and the term exposotype is already in the literature, so I borrowed it. It's actually used in metabolomics. It's analogous to genotypes. It would refer to the environmental exposures at the individual level or the exposure phenotype. Oops. So our exposome has a correlation structure. If we understand correlation, we can impute missing data. We do this all the time in epidemiology. If we had a human exposome map, that could be the linchpin for exposome imputation by defining tagging exposotypes, that is, key exposures that predict the presence of other exposures. So here's an example of an exposotype tag method. So I would argue that home or location and life stage are two of the most important tags. So if you know someone's home location, that tags air pollution, tags all kinds of climate variables. It will tag water, it will tag socioeconomic status, and many, many other exposures. If you know life stage, just someone's age, that will tag certain aspects of diet, play, activity, sports, hobbies, etc. And income, products that you use, occupation, all these are likely to be important tags. And there may be hundreds of thousands of tags. There may be some tags that turn out to be epigenetic marks. There may be some tags that turn out to be other biological factors. But these tags will actually predict the presence of other environmental exposures. And they may be time varying or life stage specific. If we understand their correlation structure, we can start to impute missing parts of the exposome. So if we only measure a subset of the exposome, we can impute the missing parts. And we can do that over both space and time. So if we'll have these cross-sectional measurements that are longitudinal, and we understand the correlation structure among the different measurements at any given time, we can also impute going backwards in time if we know the correlation structure over time. So if red is missing exposome data and green is measured exposome data, the correlation is proportional to the distance between the dots. We can actually impute the red dots if we measure the green dots. And with a temporal geospatial map of everything, we could eventually determine the exposome's correlation structure, impute the larger exposome at the individual level instead of measuring it all. And we already have the tools to do this with machine learning and multiple imputation models. So some uses of the human exposome map. So here's a simple example. I'm just going to take myself based on where I've lived. So I'm 61 years old. I was born in Okinawa in 1963 next to an Air Force base. I lived there till I was 3 years old. I grew up in an area of Detroit that they called Downriver from age 4 onward, which is right next to the Ford Rouge plant, which was at the time the largest auto factory in the planet. I'm first generation college. I went to medical school. I practiced pediatrics for 20 years. I've lived in Chicago, the Boston suburbs, Westchester, New York. I exercise regularly. But I did develop coronary artery disease and had surgery just a few months ago. However, if we have my address and link it to my life stage, we can actually start to reconstruct my lifetime exposome. So as I said, I was born in Okinawa. I was born next to Kadena Air Force Base. And if you do even natural language processing searches on Kadena Air Force Base, you'll find that Agent Orange was spread around there as a weed killer during the Vietnam War, which is roughly when I was born. And this is based on an interview of a veteran stationed at Kadena. Also, a lot of military bases have a lot of environmental contaminants. So it's probably safe to say that I may have been exposed to those common contaminants that occurred around military bases, both prenatally and in my early childhood. I moved to downriver Detroit next to the Ford Rouge plant when I was about 3 years old and lived from that age until age 25 in the same house. That's an area which is in the box, a town called Melvindale. Lower socioeconomic status, lots of air pollution in this area. So that's another exposure that I had during childhood. After I grew up, went to medical school, I started a family. I lived in the suburbs of Boston, which had relatively low air pollution, perhaps more pollen, but lots of woods and lots of green space and very good scores on neighborhood sentiment and safety index. The house I grew up in got broken into 3 times, and actually there was a shootout once in my backyard. I don't talk about that a lot, but it wasn't the safest neighborhood. Where I live now is in Westchester, New York, which is a upper middle class neighborhood. Very good air quality. It's suburban, but it's got a little more of an urban feel, which means there's a lot of traffic. So probably air quality isn't as good as when I lived in Massachusetts, but still pretty safe. But consider the contrast of my early life exposome with my adult exposome. If all I knew if I entered a study was my current address, what you know about my previous exposures is very, very limited. So I think we miss a lot because we don't ask people about their childhood place of residence and what potential environmental exposures exist. If we had a human exposome map that was both temporal and spatial, we could even start to reconstruct what happened to people early in life. And if we could do it on a larger scale, we could start to look at whether or not there are environmental exposures that are likely predicted by where you lived at particular life stages that actually predict your health today. We could do developmental origins of health and disease research on a grand scale. So the Human Exposome Map project would enable exposome-wide association studies so that we could actually look at life stage and the exposome using the Human Exposome Map. And as I mentioned, it would enable, I think most importantly, developmental origins of health and disease studies because the maps would capture time variation, allowing the imputation of the past exposome, and would allow visualization of the co-localization of health and exposure, thereby informing health disparities. Seems to be a delay. Sorry, my computer had a delay. So a human exposome map to reconstruct the past, I would argue, is the Rosetta Stone of exposomics, developmental origins of disease, also gene-environment interaction research and health disparities, and is really the grand challenge of the 21st century. And this is a summary of the protocol of the progress cohort that Mara and I started many, many years ago, almost 20 years ago. We collected data in pregnancy, and we've seen children in 2-year intervals up until age 15 to 16. We actually have the ability to actually use imputation methods to take exposures measured at age 10 to try to impute measurements at birth. We'll know what the truth is because we did those measurements at birth, and we can see what factors actually predicted. So this is something I think we need to work on going forward. So in summary, everything can be mapped everywhere all at once, but also across time. This includes biomarkers, both in blood and urine, other types of biomarkers, biological factors such as cytokines, includes climate, air pollution, green space, noise and light, socioeconomic factors, social media, buildings, green space, businesses, real estate data. All these things can be mapped, as well as health, infections, depression, cancer, and other chronic diseases. And these maps can be temporal. Temporal geospatial maps can be the foundation by which we impute the past exposome. And there's lots of incentives for doing this. We're all health researchers, so it's important to have a research infrastructure tool, which I think this talk actually is the bulk of. And that would be important for public health, to prevent disease, respond to outbreaks, predict and respond to disasters. There may be clinical uses. Maybe we can understand some treatment failures or prolong health after surgery or with cancer or heart disease. But I think probably the biggest driver is going to be commercial usage. Real estate, urban planning, diagnostic tools, tracking health trends, apps for environmental exposures. I think people want this information, apps that help consumers to choose products. The commercial usage actually is likely to be the greatest incentive to fund an exposome map project. And it's something we have to really embrace because I think this is the way it will actually happen. All the other factors, sadly, are less important to most governments than the commercial viability of an idea. And if you don't think that a geospatial temporal map is important, don't tell that to Google or Zillow, because many of these companies are already monetizing these types of maps. So everything in this talk actually already exists. So this is a quote from an author, science fiction author, William Gibson. He wrote a story called Neuromancer, on which the Matrix movies were actually based. And this is a quote from the book. The future is already here. It's not very evenly distributed. What he means by that, all the tools that exist that we need to advance our society already exist. We just don't use them in an integrated way. And I would argue that that's really what we need in exposomics to create this human temporal spatial exposome map. We don't need game-changing technology. We just need a big picture data architecture project that collates and integrates existing resources and builds a coalition of cross-disciplinary researchers dedicated to exposome research. Well, thank you. And these are all the people that have helped me on this idea over the years. We've had several meetings where we've had discussions about this. You can see Mara is near the top of the list as well. And again, another plug for my exposome blog. So thank you.